Have you ever just stopped and wondered what really makes you, you? From your eye color to why you can or can't handle spicy food? Well, it turns out the answers are written in your DNA. Today, we're going to dive deep into the incredible world of human genetic variation. We'll look at the tiny, tiny differences that make every single one of us unique, and we'll see how understanding our own genetic blueprint can completely flip some of our biggest ideas about who we are.
I mean, it's the biggest question of all, isn't it? We're all human, but at the same time, we're all so different. So what is it, deep down in our biology, that creates all of this amazing diversity we see every day? So yeah, the answer, of course, is in our DNA. But this isn't just about what our genes do say. We're also going to unpack what they don't say. And trust me, you might be surprised to find out that a lot of what you think you know about genetics, well, it's not quite the full story.
And this, right here, is the perfect way to frame our whole conversation. This genetic variety we're talking about, it isn't some kind of mistake or a bug in the system. It is the system. It's literally the engine of life, the source of our strength as a species. It's completely natural and absolutely essential.
Okay, so let's get into the nitty-gritty. How do these differences actually pop up in our DNA? We're going to start with the absolute basics, the most common kinds of variations that form the building blocks of our entire genetic blueprint.
All right, so the most common type of genetic variation is something called a single nucleotide polymorphism, or for short, a SNP. The easiest way to think about it is this. Imagine your DNA is a gigantic encyclopedia. A SNP is like a single-letter typo in one of the words, and if that specific typo shows up in at least 1% of the population, we give it the official name SNP.
So the big question is, are these SNPs good or bad? Well, that's not really the right way to look at it. A much better way to think about them is like different kinds of shoes. Are hiking boots better than flip-flops? It depends. Are you climbing a mountain or going to the beach? An SNP is just a difference, and our default position should be that it's neutral, unless we have really solid data that tells us otherwise.
So how in the world do scientists even begin to spot these differences in genomes that are billions of letters long? Well, one really cool tool they use is called a dot plot. If you take a DNA sequence and compare it to itself, you get this beautiful, perfect diagonal line. It's a super simple, visual way to confirm you've got a perfect match.
But what happens when the genomes don't match perfectly? See that little break in the diagonal line? That gap is the visual smoking gun. It tells you a chunk of DNA has either been added or, in this case, deleted. Scientists call this an indel, and it's another really common way our genomes can vary.
Okay, so we've got SNPs and we've got indels, our different shoes. But sometimes these changes are definitely not neutral. So when do these tiny little variations start to have major life-altering consequences for our health? Let's look at a couple of really powerful examples.
Check this out. It's a really intense clinical story. In some cases of eclampsia, which is a really dangerous condition in pregnancy, the root cause is a tiny insertion of just 14 DNA letters in the fetus's genome. That tiny change sets off a chain reaction where a key protein just vanishes. And the result? The mother's immune system literally starts attacking the fetus. It's a tiny change with an absolutely enormous impact.
And here's another jaw-dropping example of a different type of variation. In Huntington's disease, which is a truly devastating neurological disorder, the genetic error is basically a stutter. A three-letter sequence, CAG, just repeats itself way too many times. A healthy version of the gene might have around 11 repeats, but the version that causes the disease, it has 44 or more. That number right there tells the whole story.
Now, we're going to get into a concept that is way more common than you'd think. Most of us just assume that every cell in our body has the exact same set of genes. But what if that's not true? What if you had patches of cells with different genetic codes? Well, that's a real thing. It's called genetic mosaicism, and it's estimated that maybe 10% of us are walking around like this.
And the absolute best way to picture this is with a calico cat. The genes for fur color are on the X chromosome. In female cats, one of their two X chromosomes gets randomly switched off in every cell early in development. So depending on which one gets turned off, you get a patch of orange fur or a patch of black fur. Each patch is literally a different set of active genes. That's mosaicism in action.
Okay, so let's apply this to a real-life medical mystery. Imagine this. A family has two children, and both have a rare, dominant genetic disorder. But here's the puzzle. Neither of their parents has the disorder. And when doctors test the mother's blood, her genes look totally normal. So
If she doesn't carry the mutation, how on earth could she pass it on to two different kids?
And the answer, incredibly, was mosaicism. The DNA test was done on her blood cells, which were normal. But her ovaries contained a population of cells that did have the mutation. This means she was producing eggs with a totally different genotype from the rest of her body. It's just a stunning example of how weird and complex our genetics can be.
So we've got a handle on how genetic variation works. Now we're going to take everything we've learned and use it to tackle one of the biggest, most persistent, and frankly, most harmful misconceptions in science and society, the idea that human races are a biological reality.
This quote from a doctor in a neonatal ICU just cuts right to the chase. When you're making life or death decisions for a tiny baby, the social label of race is useless. What actually matters is the patient's real genetic background. And as we're about to see, those two things are not the same at all.
But this confusion between race and genetics has led to some really misguided decisions. For instance, the FDA actually approved a drug that was specifically marketed to patients who identified as Black. Now, this whole approach was built on the flawed assumption that race is a meaningful and reliable category for medicine.
Okay, so let's just look at the data. On this chart, every single dot is one person's genome. And the closer two dots are, the more genetically similar those two people are. Right now, all these blue dots you see represent the entire range of genetic diversity found within just one group of people that society might lump together as a single race. Now, let's add the data for a second group of people from a different so-called race. And look at that. You see what happens? There is a massive area of overlap. You literally cannot draw a clean line to separate these two groups. Genetically, that line just doesn't exist.
And now for the third group. It's almost a total wash. The overlap is nearly complete. This visual makes the single most crucial point absolutely undeniable. You will consistently find more genetic diversity within any one of these so-called racial groups than you will find between them. The categories we call race simply fall apart when you look at the genes. And here's a super simple analogy to make this crystal clear. Imagine you're a vet. Would you treat all the black dogs the same and all the white dogs the same just based on their fur? Of course not. That's absurd. A black pug and a black Labrador are totally different animals. Skin color, just like fur color, is one superficial trait out of millions. It's not a meaningful way to group individuals.
So, what's a better way to think? Let's take sickle cell disease. It's often mislabeled as a black disease. That is flat out wrong. What it actually is a genetic mutation that gives people resistance to malaria. So where do you find it? You find it wherever malaria has been a major problem. That includes parts of Africa, for sure. But it also includes populations all around the Mediterranean Sea, in the Middle East, in India, among people from all sorts of ancestral backgrounds. The real link isn't to race, it's to geography and environment.
And all of this leaves us with one last massive question. We've seen that race is a really powerful social construct, but it is not a biological reality. So the future of good, effective medicine can't be based on these superficial, flawed labels. The real challenge, and the incredible promise, is to build a future of medicine that finally looks past the label and treats what's actually there, the unique genome of every single individual.